कधी दाबायचं? इकडे काहीतरी करत होते का ते? हे नुसतं दाबायचंय आणि काय होतं मग? हा हा दाबलं. हा आता परत काय
इकडे हा जो वन ड्राईव्ह आहे ना,
कुठे कुठे?
क्लाउड इथं आलं बघा, सेव्ह ऑन युअर वन ड्राईव्ह. आता तुम्ही जेव्हा तुम्हाला घ्यायचं तेव्हा ते फक्त बटन दाबा, काय करायचं नाही बघा, आपल्या ह्याला होतं सेव्ह.
असं म्हणजे पीपीटीत काय हॅलो, एम आय ऑडिबल? हॅलो, अरे आवाज ऐकू येतोय का?
सर आवाज ब्रेक होतोय.
काय आवाज केले की काय माझं मोबाईलचं दिलंय का मोबाईल. हॅलो, आवाज ऐकू येते का बाळानो? हॅलो, एम आय ऑडिबल? तर घेऊया ना व्यवस्थित, अदरवाइज वी विल वेट फॉर द इलेक्ट्रिक सप्लाय. येस. आशिष
येतोय सर आवाज.
घाडगे, शिवानी. असं काय तुला काय आता येतोय का? बर, बघूया, शेअर करतो की. असं नाही काय होतं. कुठल्या डिव्हिजन मध्ये? मग जीचं लेक्चर चालू होते का सर? अगं ते आपला ग्रुप तयार केला की जेव्हा लाईट गेली आई मला अरे पीपीटी दिसत रे बाळा. ऍड करून मेसेज टाकले की माझ्या लक्षात.
नाही दिसत सर पीपीटी.
Yes, students, see that you join quickly. Only 16 of you on the screen. The previous lecture, due to the electricity problems, there was no electricity connection, therefore the first lecture was not there. We could not conduct the first lecture, but now as the electricity connections are again on and we have the internet facility also, I will be starting with my next lecture. So see that you join very quickly. I will be able to conduct the lecture. So yes, just 2 to 3 minutes more. See that you very quickly join the lecture. एक साधारणतः एक चंद्रमणी आठवणी आता सुरूच करते मी मुलं जॉईन होतात अजून एक 5 दरम्यान रिकॉल करते. So students, can you see the screen please? Yes. Can you see the screen? So this will take approximately 5 minutes for your answer, isn't it? So just a second from now, I had shared a screen to you. I'm asking whether I'm audible and whether the screen was visible. So am I audible to you? Please be muted off from your side, someone. A lot of people are there and everyone have joined, okay? But over here, I can see only 31 and 32 of Vaishnavi Pawar, okay? So please, Aditya Chikane, Deepa Nalaude, Bharati Yalmar, from your side, let me know whether I'm audible and whether...
Yes, ma'am.
Yes, and whether just a few seconds ago I had shared the screen, whether the screen is visible to you.
Yes, ma'am.
Okay, so just see, we are back to the screen. Is it visible?
Yes, ma'am.
Okay, so fine. We will be now starting with a new unit, which is Unit 1 from your virology paper. So this will introduce to you after your first year where you had known some basics of virus, some basic properties of viruses. Over there, you have seen that what do you mean by viruses, how small these agents are, and some of the characteristics of viruses. You've also seen that viruses, the microbiologists say that viruses are the borderline between living and non-living. That is, they share the properties of living organisms, as well as they also have some properties of the non-living beings. So at FY/BSc level, you have seen only about some basic concepts about the viruses. So at SY, we do not have much about viruses in our syllabus incorporated, and it is in the TY that you have to deal with a separate paper on virology. So here we come with Unit 1, which deals with the introductory part of virology. So we will start with the virus definition. So a virus is a very small, non-cellular parasite of cells. Its genome is composed of either DNA or RNA, and this genome, it is enclosed in a protein coat. So this is the definition of a virus. Very small, naturally smaller than bacteria. They are non-cellular parasites. We will see this term. What do you mean by non-cellular parasites? So over here you can just understand that they are parasites of the cells. That is, they live on the cells, parasites of the cells. They, you can say, take all the nutrients from the cells. They cannot, these viruses, they cannot manufacture their own liveliness, their own nutrients, their own constituents independently. They require the cells for the reproduction. So this is meant by parasites of the cells. They have no independent existence. They replicate only when within the living cells. Outside the living cells, they may be present for years, for centuries also. But without the cells, they do not show any signs of their liveliness. So we can say that unless and until the particle, or unless and until a constituent interacts with the cells, it is said to be non-living. So in that aspect, when viruses are outside the cells, as they do not have the cellular machinery to replicate, to reproduce, or to induce any changes within the cells, outside the cells, we can say that these are the non-living creatures, non-living particles. So here goes the definition that viruses are very small, non-cellular parasites of cells. They possess a genome which is composed of either the DNA or the RNA. We will shortly see how the genomic constituents of the viruses are. And this DNA or the RNA, it is enclosed in a protein coat. So as to now, you can just remember that viruses, they have a protein coat within which you have the genetic material. And this genetic material and the protein coat also, it differs from virus to virus. And there are many types of viruses, many subtypes of viruses, which are differentiated depending upon the constituents of the protein as well as the genetic material. So the next point, that is the nature of viruses and the discovery of viruses. So firstly, it was Martinus Pijering in Holland and Dmitry Ivanovsky. They were the first ones in the 19th century to discover the TMV virus. So TMV is tobacco mosaic virus. This virus, it was discovered by these two scientists where they carried out a very simple experiment, that is the diseased tobacco plants. So tobacco plants are diseased, meaning thereby the healthy plants, they have very healthy leaves without any spots or without any, you can say, physical damage to the leaves, to the plants. But in cases where the tobacco mosaic virus infects the tobacco plants, there are many symptoms seen on the leaves where we can observe certain patches where chlorosis has been occurred, then the decoloration of the leaves occur. At some points, the upper surface and the lower surface is also damaged, and it looks, you can say, having different types of cankery portions on the leaves, which itself differentiates the diseased leaf from the healthy leaf. So the appearance of this, you can say, tobacco leaf, it is not good. Uh, it is not a good sight. So, and this virus, of course, when infects the tobacco leaves, it is not a single leaf that is affected. It is the total crop in the field that is affected, and therefore this results in burning of the leaves, and the whole tobacco plant, it is destroyed by these viruses, because the leaves may be you can say they are not healthy, as well as at a later stage of infection, the viruses, they feed upon these cellular parts of the leaves, and they are now no more in a healthy condition to be sold. So this, in the 19th century, was observed in the plants, that the tobacco plants were infected with some type of an agent. And an experiment carried out by Beijerink was that, and Ivanovsky, of course, was that they crushed the leaves, infected leaves, and passed them through the filterable filter, the bacterioproof filter rather. So the filtrate was injected again to the healthy tobacco plants, which was found to be infected with the viruses. So from this experiment, these two, they drew a conclusion that there was something in the filtrate which was smaller than the bacteria, and the agents which were present in the filtrate were responsible for the diseases in the tobacco plants. The term virus, which literally means poison, virus means poison. So this term virus was also coined by Beijerink, virus. Later, after this discovery that there are some agents which were named as virus by Beijerink, after this discovery, again independently, Frederick Loeffler and Paul Frosch, they discovered a similar type of agent which was responsible for the foot and mouth disease in animals. So animal, you can search over here, foot and mouth disease, that is the feet of the cattle as well as the mouth. They showed some symptoms that are such as swelling of the feet, you can say infection of the feet and the mouth itself. So this disease, which is known as foot and mouth disease, the name itself indicates that the cattle was infected, the foot and the mouth of the cattle was infected, and therefore the disease, foot and mouth disease, was also found to be, you can say, experimented by these two scientists, and found that the similar agent which was very smaller than bacteria is responsible for this disease. So over here it was, and of course again one more scientist, that is Walter Reed and James Carroll, they discovered that yellow fever is also caused by a filterable agents. So just see the beauty of these experiments and the work of these scientists. Just passing of you can say, the crushed material from all these disease parts of either the plant or the animal parts, just crushing of this disease parts and a simple experiment of filtering this crushed part through a filtrate which was bacteria proof, which did not allow the bacteria to pass through the filter. The filtrate that was obtained, it contained something. All these scientists, they concluded that this filtrate did not contain the bacteria, but they contained those agents which were smaller than bacteria, as a bacteria-proof filter was used. This filtrate, it had agents which are smaller than bacteria, and these agents are responsible for causing different types of diseases in plants as well as animals. So, very simple experiments bringing down into or focusing on an agent which was responsible, which was smaller than bacteria and was responsible for causing certain, you can say, disease conditions in plants as well as the animals. So the credit goes to all these scientists who worked and focused on the particles which were responsible for the disease conditions. Now we'll move on to the next point, that is the size of these viruses. So what is the size of the viruses? From the experiments of these scientists, Beijering, Loeffler, all these scientists which we have seen in the first slide, they came to the conclusion that they were quite smaller than the bacteria. But what is the exact size and in which size they are measured? So virions, viruses, virions, this is a new term, maybe a new term to you, that is viruses with DNA and a protein coat, intact viruses, infectious viruses, these are known as virions. So the viruses are normally measured in nanometers. Now what do you mean by nanometers? 1 nanometer equals to 10 raised to minus 9 meters. Okay? एका मीटरचे एवढे भाग केले तर त्याच्यातला एक भाग, if it is divided into so many divisions, या एका मीटरचे एवढे डिव्हिजन्स केले तर त्यातल्या एका डिव्हिजनची किंमत म्हणजे 1 nanometer. So it is a very, very small scale. So here is a picturization to give you the, you can say a visualization of the size of the viruses. So here you can see the virion of the herpes virus, which is fairly large as compared to the smaller virus than that. But see, if you see the herpes virus, it is a fairly large virus, but it is 10 million times smaller than a large balloon. Okay, it is smaller 10 million times than a smaller balloon. While this balloon to which the size of this herpes virus is compared, balloon itself is smaller than Earth by 10 million times. Okay, so did you get the, you can say, picturization or the concept of the size of the viruses? Okay, if this is considered as the virus, this virus, it is 10 million times smaller than the balloon. And this balloon, it is 10 million smaller times than that of the size of the Earth. So this is about the size of the viruses, which is measured in 1 nanometers, which is equal to 10 raised to minus 9 meters. Okay. So viruses, they cannot be seen under the light microscope. We usually observe in our microbiological lab. You've seen at the FY and SY level that microorganisms can be seen under the oil immersion lens, isn't it? We have done many staining procedures. The negative staining, special staining, gram staining, cell wall staining, so on and so forth. So in these staining techniques, you all have seen microorganisms. At the SYBAC level, you must have seen certain pathogens which we have identified at the syllabus of your SYBAC. So the point here is the microorganisms can be seen under the oil immersion lens, but viruses which are very smaller than the bacteria, they cannot be seen with the light microscopes. They can be seen only with electron microscopes. So what is an electron microscope? We have seen at the SYBAC, at the FY BAC level. So you can see the scientist or the lady working with this instrument, electron microscope. So for observing the viruses, you need this instrument, which is an electron microscope. You can never observe viruses under the light microscopes or the oil immersion lenses. So we will see the range of the size of the viruses. So though the viruses are very small, it is not that all the viruses have the same range of the sizes. म्हणजे काय की थोडक्यात की एक 2 micrometers, so 2 nanometers, not micrometers, it is smaller than micrometers, 2 nanometers, 20 nanometers, 10 nanometers. So you cannot expect the viruses to be of a similar size or a size difference of only a few nanometers. That is, some viruses you can say they are 10 nanometers, some are 12 nanometers, and they have a range between 10 to 15 nanometers, and that's all. No, this is not the condition. They have a vast coverage of their sizes. Amongst them, we have two viruses. Parvoviruses, they have diameter of approximately 20 nanometers. And the next virus, that is a microbe mimicking virus, which is also known as the Mimi virus, which is isolated from amoeba. This is one of the largest virus that is seen. So this is the Mimi virus isolated from amoeba. And this is the parvovirus. Okay, so you can see the size difference. Smallest virus, which has approximately 20 nanometers, and this is the largest virus as compared to this virus. Okay, so again, this is the size of the mimicking virus. That is, it is approximately 500 nanometers. Okay, so this gives you an idea about the large cover, a large range, dimensions cover of the viruses. Small viruses are also present and larger viruses also present. Of course, intermediate size viruses are also present. So this is about the size of the viruses. It is measured in nanometers, and different sizes of viruses are present. The smallest is known as the parvovirus, and the largest up to now, it is the Mimi virus isolated from amoeba. So now we will see about virions. So just see. First, let me confirm whether the screen is visible to you. Yes, please, any one of you, please.
हो मॅम दिसतीये.
दिसतीये स्क्रीन आणि ऐकू येतंय व्यवस्थित? ऐकण्यामध्ये काही प्रॉब्लेम्स नाहीत ना? आवाजामध्ये काही फरक व्यवस्थित येतोय ना सगळीकडे? ओके. आणि मुख्य म्हणजे समजतंय सगळ्यांना? जे काही स्क्रीन चाललंय ते समजतंय? ओके. ओके. सो वी विल रिझ्यूम with the lecture. So So now just see, what are virions? I told you that virions, they are the virus particles which are infective, that is intact virus particles with the protein coat and the nucleic acids within it. These are known as virions. So virions are not cells. Like the living cells, these are not the cells because they do not contain organelles except for the virions of the arenavirus. Arenavirus असा एकमेव virus आहे की ज्याच्यामध्ये which contains the cell ribosomes that were packaged when the virions were assembled. म्हणजे हा एकमेव इन्फेक्ट करतो इंडिव्हिज्युअलला युकॅरियोटिक सेल्सला, त्यावेळेला मल्टिप्लिकेशनच्या वेळेला मल्टिप्लिकेशन किंवा असेंब्ली पार्टिकल्सची ज्या वेळेला होते त्यावेळेला असेंब्लीचा डिफेक्ट म्हणून काही व्हायरसेसमध्ये रायबोझोम्स पण पॅक केले गेले. आणि हा एकमेव व्हायरस आहे की ज्याच्यामध्ये युकॅरियोटिक रायबोझोम्स आढळलेले दिसतात. ओके, सो viruses, they are not the cells, they do not contain any cell organelles as we see in the you can say normal cells. एक सेलचं स्ट्रक्चर प्रोकॅरियोटिक असेल किंवा युकॅरियोटिक असेल तर सेल्स मध्ये बाकीचे सगळे कॉन्स्टिट्युएंट्स असतात. मग प्रोकॅरियोटिक सेल्स मध्ये तुम्हाला सायटोप्लाझम असेल, त्याच्यानंतर रेप्लिकेशन एन्झाईम्स असतील, प्रोटीन्स असतील, विटामिन्स असतील, रायबोझोम्स असतील, प्रोकॅरियोटचे वेगळे रायबोझोम्स, युकॅरियोटचे वेगळे रायबोझोम्स, आणि त्यांचे सेल्युलर रिॲक्शन्स मेनली असतील विच आर रिस्पॉन्सिबल फॉर द रेग्युलर मेटाबॉलिक ऍक्टिव्हिटी ऑफ द सेल which provide all the intermediate compounds responsible for the metabolism of the cells, so on and so forth. तो वेगवेगळे जे काही ऑर्गॅनल्स आहेत, कॉन्स्टिट्युएंट्स आहेत, मॅक्रोमॉलिक्युल्स आहेत सेल्स मध्ये, मग ते प्रोकॅरियोटिक असो किंवा युकॅरियोटिक असो. So these viruses do not contain any type of organelles. All that is present in the viruses, it is only a protein coat within which they have the you can say the genetic material. So we will shortly see what type of a genetic material they have. So virology, it is therefore concerned with very small particles, though often with very large numbers of those particles. A concentrated suspensions of virions may contain 1012 virions per ml. A single virus infected cell might produce 105 virions. A person infected with HIV may produce 1011 virions per day. म्हणजे काय की व्हायरॉलॉजी किंवा व्हायरसेस, व्हायरियॉन्स जरी असे पार्टिकल्स आहेत की ज्यांच्यामध्ये ऑर्गॅनल्स नाही, पण तरी सुद्धा इन व्हायरॉलॉजी वन्स in this branch of science, branch of microbiology related with the everything with the viruses. So here we are concerned with these virions, small particles, but once within the cell, ज्यावेळेला हे viruses cell मध्ये जातील, मग plant cell असू दे किंवा eukaryotic cell असू दे, they reproduce and they produce large number of those particles by utilizing the cellular machinery. Okay, so once within the cell they reproduce, and the concentrated suspensions of the virions within the cells or after the burst of the cells, it may contain many virions. That is, as it is during infection or when the infection takes place of a cell, a single virus infects, a single type of a virus infect, you can say. And this virus then reproduces, and the suspension which is obtained after the reproduction phase of the virus is over, it is tremendously large. So from one virus, depending upon the reproduction of the viruses. From one virus we can have 1012 virions per ml. एका ml मध्ये एवढे व्हायरन्स असू शकतात. एवढ्या फास्ट रेटनी व्हायरसेस हे मल्टिप्लाय होतात. Viruses infected cells, they may have 105 virions. Or HIV patients, it has been studied that per day, in a day, it has 1011 virions in a day. म्हणजे एका व्हायरस पासून एका सेल मध्ये एवढे व्हायरियॉन्स 24 तासामध्ये तयार होतात. So this is due to the reproduction of the viruses after getting the ready-made cellular constituents, which is made use of for its own reproduction by the viruses. So next, viruses are parasites. So this is again the point of discussion where the viruses differs from the cells in a way in which they multiply. A new cells मध्ये ज्याला eukaryotic असेल किंवा prokaryotic cell असेल, a new cell is always formed directly from the pre-existing cell. Okay, but in case of viruses, a new virion is never formed directly from the pre-existing virion. New virions are formed by the process of replication, which takes place inside the host, and it involves the synthesis of components followed by the assembly of the virions. हे तुम्हाला मी मगाशी सांगितलेलं आहे. Okay, so the next point. So I told you that just a minute. So about the virions. So viruses therefore are the parasites of the cells, and they are dependent on the host for the multiplication, the requirements for multiplication. So this point has already been covered. So what do they require from the cell? Viruses ज्यावेळेला इन्फेक्ट करतात एखाद्या सेलला, ते सेल्स त्यांचं स्वतःच असं व्हायरसेसचं काहीच नसतं. काय असतं तर only the DNA or the RNA, that is the genetic material, and a protein coat. Nothing is present, nothing else than this. In some viruses, you can have certain enzymes, along a small amount of enzymes in this protein coat, along with the genetic material. But as you have seen in the adenovirus, these contain also some ribosomes, which has been taken as a misassembly from the eukaryotic cells. But besides this, viruses have nothing so that they will be able to live, or they will be able to continue their metabolic and catabolic activities. They will be able to take the nutrients. They will be able to reproduce on their own. Nothing of such sort is present within the viruses. लक्षात येतं सगळ्यांच्या. तर viruses हे सेलच्या बाहेर जोपर्यंत त्यांना अप्रोप्रिएट सेल मिळत नाही आणि जोपर्यंत ते आतमध्ये जात नाहीत, लिव्हिंग सेल्सच्या आतमध्ये जात नाहीत, तोपर्यंत दे आर नॉन लिव्हिंग ऑब्जेक्ट्स. लक्षात येतंय? कारण का नॉन लिव्हिंग आपण का म्हणायचं? कारण हे जे व्हायरसेस आहेत ते व्हायरसेस सेलच्या बाहेर, दे आर पॅरासाइट्स. लक्षात घ्या, पॅरासाइट्स कुणाला म्हणायचं की दोज ऑर्गॅनिझम्स विच कॅनॉट मॅन्युफॅक्चर देअर ओन food material, their own metabolic activities. They cannot reproduce on their own. So these are non-living things. So in this aspect, viruses are non-living outside the cell. They do not possess any of the cellular machineries which will help them to carry on their independent functions, independent satisfaction of the independent nutritional requirements. They cannot independently carry on any biochemical reactions required for their catabolic and metabolic activities. They cannot carry out the reproduction. They cannot multiply outside the living cells. So when once within the cells, viruses, they depend on the host cells for their requirements. On which aspects they depend, on which you can say macromolecules they depend on amino acids, nucleosides. They depend on protein synthesizing machinery, they depend on the ribosomes. They depend again on the DNA replicating enzymes of the host. They require energy from the host. So, when once within the living cells, these viruses are so smart, they have the genetic information within the DNA or the RNA so that they will, you can say, alter the environment of the host and utilize all the macromolecules of the host, enzymes of the host, energy of the host to multiply its own DNA and to produce the, you can say, proteins or the capsids and the envelope of the protein, which is similar to its own. In this manner, by the use of all the cellular machinery, all the cellular macromolecules, all the cellular contents, constituents, the virus DNA, it conquers the host cells and it reproduces or it produces an array of macromolecules which are required for its assembly, from which after assembly it produces a very high yield of a crop which has similar units. We can say it reproduces from one virus to thousands of viruses. So, in what way does the virus manage to do this? So, a virus modifies the intercellular environment of the host in order to enhance the efficiency of the replication process. या सगळ्या गोष्टी आपण व्हायरोलॉजी मध्ये बघतो. रिप्रोडक्शन ऑफ डिफरंट टाइप्स ऑफ व्हायरसेस आपण बघणार आहोत. त्याच्यामध्ये हे सगळं म्हणजे एक एक्झाम्पल व्हायरसेसचं घेतलं तर आपण T4 बॅक्टेरियोफाजचं रिप्रोडक्शन फॉर एक्झाम्पल. तर बॅक्टेरियामध्ये जाऊन T4 बॅक्टेरियोफाज बॅक्टेरियल सेल्सचे जे काही मॅक्रोमॉलिक्युल्स आहेत त्याच्यात मॉडिफिकेशन कसे करतात आणि या मॅक्रोमॉलिक्युल्सला अल्टर करून ते स्वतःच्या उपयोगासाठी कसे डायव्हर्ट करून घेतात, या सगळ्या गोष्टी आपण मॉलिक्युलर लेवलला व्हायरोलॉजीच्या पेपरमध्ये पाहतो. सो roughly you can remember that a virus modifies the intracellular environment of the host in order to enhance the efficiency of the replication process. Modification might include the production of new membranous structures, reduced expression of the cell genes, or enhancement of the cell process. Some large phages encode proteins that boost the photosynthesis in the cells of the photosynthetic bacterial host. Photosynthesis bacterial host मध्ये, photosynthetic bacterial host मध्ये जर काही phages, of course these phages they are very specific in their infections. म्हणजे त्या बॅक्टेरियम मध्ये, फोटोसिंथेटिक बॅक्टेरिया मध्ये ज्यावेळेला हे phages जातील त्यावेळेला हे phages they will be very specific to that photosynthetic bacteria. But after infection, in such bacterial host, these viral particles are found to enhance or boost the photosynthetic activity of the bacterial host. And this boosting probably results in a greater yield of the viruses from the cells. So these are some of the mechanisms by which the viruses, they play their tricks rather. They extract whatever they want from the bacterial cells or from the eukaryotic cells. And this is how, by the use of the cellular machinery, the cells which they infect, by the use of the cellular machinery, they reproduce themselves. So the next point is the viruses, they have genes. So as I told you in the previous points, that is viruses, very small in structure, measured in nanometers, and they are composed of only two parts. That is the core of the viruses, which consists of, you can say, the genetic material, which is enveloped, or this genetic material in some cases may be protected by nucleoproteins, and this you can say core, which contains the genetic material, the core is then covered by proteins, and there are different types of proteins. There may be you can say glycoproteins also involved, which forms the covering portion. So you can just imagine at the prima facie, you can imagine the structure of a virus just like a ball, a rubber ball, which usually children play with. So of course the viruses always do not have a shape of a ball. Different types of shapes we have of the viruses, which we'll be seeing in the coming lectures, the architecture of the virus capsids. So just to imagine, you can imagine it, the viruses as a ball. The outer covering, the rubber covering, can be imagined to be the protein coat of the viruses, and the inner hollow portion of the ball will contain the nucleic acids. And these nucleic acids may be of different types. So there are four possibilities of the virus genome. म्हणजे व्हायरस जीनोम जो आहे तो जीनोम आत कोर मध्ये कसा असू शकतो किंवा कोणत्या प्रकारातला असू शकतो. So it is composed of the following. It may be a double stranded DNA, or it may be a single stranded DNA, or it may be a double stranded RNA or a single stranded RNA. And again, there are many types here also. There may be palindromic sequences, the RNA or the DNA. They may be again, you can say, they have within themselves different type of shapes depending upon the sequences of the RNA or the DNA. Some viruses have a pan-handled sequence or pan-handled shape. Pan-handle itself explain. Pan म्हणजे तवा. तवा जो आहे म्हणजे दांड्याचा जो तवा असतो तसा शेप असणारे डीएनए असतात, सेगमेंटेड डीएनए असतात. तर अशा प्रकारचे किंवा आरएनए असू शकतात. तर अशा प्रकारचे वेगवेगळ्या फॉर्म मधले डीएनए, वेगवेगळ्या टाइप्स मधले डीएनए आपल्याला इथे दिसून येतात. ओके, सो व्हायरसेस मध्ये. सो आय थिंक it's approximately 1:05. Okay, so over here, I think that I should stop over here, because the next lecture or the next you can say part of your, yes. Yes, all of you. Am I back again? Can you hear me? ऐकू येतंय सगळ्यांना? इकडे लक्ष द्या. लगेच तुमचे किती संख्या लगेच किती रोडावली बघा. 38 जण फक्त राहिला तुम्ही. दिस इज नॉट अ गुड साइन. कोणीतरी संवाद साधा माझ्या बरोबर. येस, टुडे नो चॅटिंग इज देअर.
हाँ मैम.
Yes. So have you understood the introductory part of the viruses?
हो मैम, समझ ले.
Okay. So why are the students in a hurry to go from the screen? एनिवे मला तुम्हाला काय सांगायचं आहे की याच्यानंतर एक प्रॅक्टिकलचं सेशन होईल, व्हिज्युअल प्रॅक्टिकल्स. जसं आपण गेल्या वेळेला डीएनए अगारोस जेल इलेक्ट्रोफोरेसिस पाहिलं होतं, हो की नाही? तसं एक प्रॅक्टिकल होईल. येस, कोणाला काय बोलायचंय का? तर आता लेक्चरचा पार्ट झाला माझा. आता मी व्हायरॉलॉजी मधलं लेक्चर घेतलं. जस्ट गिव्ह मी 5 to 10 minutes मोर. एक 5 ते 10 मिनिटांमध्ये आपण परत रिजॉईन होणार आहे व्हिज्युअल प्रॅक्टिकलसाठी. तर प्रॅक्टिकलचं पोर्शन आपण इथे घेणार आहोत. तर प्रॅक्टिकल्स तुम्हाला स्क्रीनवरती दाखवले जातील. त्याच्यासाठी तुम्ही परत रिजॉईन व्हा किंवा स्क्रीनवरती राहिला तरी चालेल. बट आय विल बी बॅक अगेन विथ द प्रॅक्टिकल सेशन. डू यू फॉलो? येस, ऑल ऑफ यू. आता काय 2 लेक्चर्स होऊन संपलेलं नाही आहे. तिसरं लेक्चर जे आहे ते प्रॅक्टिकलच्या संदर्भात असेल. तर प्रॅक्टिकलच्या संदर्भात आपण एक 5 ते 10 मिनिटात परत आपण भेटणार आहोत. तर लगेच घरी जाऊ नका किंवा डिस्कनेक्ट होऊ नका. ओके, सो
ओके मिस.
okay, so I will be back with the practicals.
तिथं टॅब म्हणायचं क्रॉप टॅब.
Your entire screen.
एंटायर स्क्रीन टू टॅब म्हणा. जी आता आपण युट्यूबची तुम्ही काढली ना ती त्याला ओके करा. स्पोर्ट्स टीव्ही सेकंड शेअर तिथं दिसेल मुलांना. नंतर सुरू झाले सांगितलं थोडस हे करून ठेवत ना पॉज करायच्या.
Pauses केलेलं आहे. Students, can you hear me? Yes?
Yes, ma'am.
Okay, so just see, we have a new practical for you at the TY-BSc level, which is known as spore staining, and this will be shown to you. Spore staining by Schaffer and Fulton's method. So just see, we have seen about the spore staining procedure, isn't it? Yes, all of you? Can you hear me? Yes? ऐकू येतंय का? शिवानी जाधव, पोस्ट ट्रेनिंगची प्रोसिजर आपण आता बघितली, सगळ्यांना दिसली का ती? ओके, हॅव यू सीन इट? दिसली का सगळ्यांना स्पोर्ट्स ट्रेनिंगची प्रोसिजर? ओके, आणखीन आपण दुसरी बघूयात. So, can you see whatever is there on the screen? Screen दिसते का तुम्हाला? Yes? All of you?
नाही दिसत आहे मॅम.
स्क्रीन दिसत नाहीये. ओके.
मॅडम काय केलं?
जरा थांबायचं काय. दिसते का स्क्रीन सगळ्यांना? येस.
नाही दिसत मॅडम.
दिसत नाहीये?
दिसते मिस.
दिसतंय ना?
हा दिसतीये आता.
ओके ओके, जस्ट सी ना. आवाज काय असा येतोय पुन्हा? Not so much to be in hibernation, but ओके, हे पहा इकडे लक्ष द्या. ऐकू येतंय मी काय म्हणते ते? जरा थोडा अजून व्हॉइस प्रॉब्लेम आहे त्या व्हिडिओला, आपण दुसरा अजून एक बघूयात.
Hi friends, in this
So just see, you will be able to share, you will be able to see the screen now. Yes, is it visible?
Yes miss.
Okay, so just see now, by both the methods how spore staining is done.
What is its structure and how it forms? If you do not know, please do.
Okay, just the audio quality is not good, therefore I am switching off. I will unmute and you just see how the procedure is done. Okay. So endoscope staining carried out by two methods, that is the Schaeffer and Fulton's method. Here there is a spelling mistake. This is the right spelling. Schaeffer and Fulton's method, जी आपण मगाशी बघितली. It is a primary stain malachite green. It is forced into the spore by steaming bacterial emulsion. Malachite green is water soluble. It has a low affinity for cellular material, so vegetative cells may be decolorized with water. Safranin is then applied to counter stain, which have been any cells which have been decolorized. At the end, the staining process, vegetative cells will be pink and the endospores will be dark green. So this is about the Schaeffer-Fulton's method that we have seen in the in the first lecture. So this is about the malachite green and the decolorizing agent. Counter stain safranin. So this is the procedure that we have seen in the last video. Take a clean grease-free slide, make the smear using sterile technique, add and heat fix the organism on the slide, cover the square of blotting paper and fit it into the slide. Saturate the blotting paper with malachite green for 5 minutes steaming. Keep the moist paper, adding more dye as required. It may be steamed in a container of boiling water. Wash the slide in tap water. Next, counter stain with 0.5% safranin for 30 seconds. Wash with tap water, blot dry, and examine under microscope. Endospores are bright green, vegetative cells are brownish red to pink. So depending upon the suspension used, organism used, you will see the spores, either middle of the cell, at the end of the cell, so on. So you can see how this appears after staining by Schaeffer and Fulton's method. So by Dorner's method, this is the next method of spore staining. So you have the carbol-fuchsin stain, which is known as Ziel-Nielsen carbol-fuchsin. So the method that is given, decolorizing agent, that is acid alcohol, counter stain, that is nigrosine. This is the method we perform in our laboratory. Donner's method. So take a clean grease-free slide, make the smear using sterile technique, and cover the blotting paper to cut the smear of the paper, smear on the slide. So saturate the blotting paper with carbol fuchsin and steam it for 5 to 10 minutes. Keep the paper on the moist and add more dye as required. Don't dry the slide. Remove the blotting paper and decolorize the film with acid alcohol for 1 minute. Rinse it with tap water. So after that, dry the slide and add a drop of nigrosine over one end of the slide and make a thin film of the stain over the smear with the help of another slide. Allow the film of nigrosine to dry, and after air drying, observe the slide under water. So how do the cells appear? These cells appear as colorless endospores. As colorless, the endospores are red and the background is black. So this is how the spores appear after staining with the Donner's method. Endospores are red, colorless bacterial cells, and the background, it is black or bluish in color. So examples of spore forming organisms: Clostridium perfringens, Clostridium botulinum, Titani, Bacillus anthracis, Bacillus cereus, D. sulfotomaculum, Sporolactobacillus, and Sporosarcina. Organisms which do not have spores: Escherichia coli, Salmonella species. Okay, so this is how both the methods of staining are seen over here. Can you see the screen? Yes? दिसतंय का तुम्हाला? स्पोर्ट्स ट्रेनिंगच्या पीपीटी जी दाखवली ती दिसतीये का? आता एका क्षणापूर्वी दाखवलेली पीपीटी दिसतीये? स्पोर्ट्स ट्रेनिंगची? आता बघा, परत मी शेअर करते. दिसतेय का?
गुगल पेज दिसतंय मॅम.
गुगल पेज दिसतंय, पीपीटी दिसत नाही हो. हे दिसतंय का? बेसिक टेक्निक्स नीड टू स्टडी बॅक्टेरिया हे दिसत नाही?
नाही, गुगल पेज दिसतंय.
ओके, बर, ठीक आहे. असं जर रिस्पॉन्स दिला तर चांगला आहे ना. ट्रेनिंग असं दिसतच नाहीये पीपीटी, अशी डाउनलोड करून उपयोग नाही. ओके, ठीक आहे. सो आय थिंक एवढं एक ट्रेनिंग तुम्हाला 2 मेथड्सनी दाखवलेलं आहे. मला वाटतं एवढं बस झालं. आजच्या दिवसापुरतं एवढं चिकार झालं. पुढे बघूयात मग आणखीन काही प्रॅक्टिकल्स किंवा यालाच धरून तुम्हाला प्रिन्सिपल वगैरे काही सांगता आलं तर नंतरच्या ट्रेनिंगच्या वेळेला सांगेल. कारण का थोडेसे व्ही लॅब वरती वगैरे आपण रजिस्टर झालेलो आहे, पण त्याला ॲडोब फ्लॅश जो ऍप्लिकेशन आहे तुम्हाला स्क्रीनवरती दिसण्यासाठी ते काही दिसत नाही किंवा ते डाउनलोड होत नाही, तर त्याच्यामध्ये प्रॉब्लेम्स आहेत. त्यामुळे आम्ही जे आहेत म्हणजे जे अवेलेबल आहे इंटरनेटवरती ते तुम्हाला दाखवण्याचा प्रयत्न करतोय आणि एक प्रॅक्टिकल्स असे आम्ही घेतोय. सो कॅन यू शो द फर्स्ट व्हिडिओ अगेन? स्ट्रेनिंगचा का? पण तो एकाच मेथडचा आहे ग, दोन्ही मेथडचा व्हिडिओ आपल्याला दिसत नाहीये ना. दोन्ही मेथडचा दिसत नाहीये. बघूयात अजून आता नेक्स्ट लेक्चरला आणखीन काय चांगलं काही दाखवता येतंय का आणि ते त्यावेळेला अवेलेबल आणि विजिबल आहे का याची आपल्याला थोडंसं पाहणी करायला लागेल. तर आता दिसतंय का हे तुम्हाला? स्क्रीनवरती दिसतंय का?
हो मॅम, दिसते.
सबस्क्राईब म्हणतंय की रे? नाही हे काय ओपन होत नाहीये बरं का. नाही म्हणजे कदाचित मला माहित नसेल, पण आय एम नॉट हे जे दिलेलं आहे त्यांनी स्टेनिंग्स हे स्क्रीन दिसतंय का ग नव्याचं?
नाही मॅम, आधीचीच आहे. स्पोर्ट्स टीम.
नाही राहू देत, आपण एवढ्यावरती आज बस करूयात. Budhale, very good. You have given a good, you can say, YouTube address, but here it is not working. You cannot get the exact staining procedure. As we have seen, we have the practicals of certain preservatives, 10% formalin and so on. So I don't know how to go exactly to this address to find out the practicals of our interest. So anyway, Prashant Budhavle is a step ahead. Okay, so I think this much is all for today. As far as your practicals are concerned, or if they are considered, we will have to again look further for good practicals. So let us try again for the next practical date. Okay, so should I leave now, all of you? Yes, we will meet again in the next lecture. Okay, तुमच्याकडचा एक वीक पॉइंट काय आहे माहितीये का? अजिबातच रिस्पॉन्ड करत नाही तुम्ही पटकन. हो नाही, नाही नाही, काही नाही, काही बोलायचंच नाही. मेसेज सोडावा तर तोही पटकन चॅट करून सोडत नाही मेसेज. तुम्ही माझं बोलणं ऐकू येत नाही का तुम्हाला? येतंय ना? चला थांबूयात का मग आता? येस, मृणाल कांबळे, साक्षी पवार, रितू शेलार. चला, ओके ओके, हॅव अ गुड डे. इमिजिएट रिस्पॉन्सेस आहे, इमिजिएट पाहिजे. तो कोणीतरी स्वतःकडचा माईक लगेच हे करायचा, ऑफ करायचा, बोलायचं लगेच. काहीतरी विचारतात की नाही मॅडम तुम्हाला, विचारल्यानंतर उत्तर अपेक्षित. ते टाईप करत बसण्यापेक्षा कोणीतरी एकाने बोललं आणि कॉन्व्हर्सेशन साधलं तरी होईल. ओके, ठीक आहे. फॉर प्रॅक्टिकल्स आय थिंक वी विल हॅव टू फेस सिमिलर प्रॉब्लेम्स, बट देन with this we will shift to some at least we will be able to visualize at least partly some of the practicals. Okay, let's hope for the best in the coming lectures of the practicals. Routine lectures we are doing it, but we are today at least we have a problem with our practicals. Okay then, so see you in the next lecture, which will be tomorrow in the morning 11:30. Okay, So, okay, goodbye. Good day.